Hi everyone, my name is Maria Aristova. I'm a fourth-year graduate student in the Department of Biomedical Engineering, and today I want to share with you some work that I've been doing on measuring blood flow in the brain using 4D Flow MRI. So before I show you a lot of images, I want you to see what I'm talking about. Here you see a brain, and the blood supply to the brain shown is the arteries, shown in red. They bring oxygenated blood to the brain, and the veins, shown in blue, carry it back. The arteries have really fast flow, and the veins have really slow flow, and in between them are a network of small pathways called capillaries where the oxygen gets deposited into the cells. In a disease called brain arteriovenous malformation, or AVM, there is a connection that directly connects the arteries to the veins through a tangle of vessels like the one you see here. And that connection can cause symptoms like seizures, strokes, or headaches for patients. And so treating them is a big problem, but it's also challenging because these formations can happen anywhere, and so they're very complicated in their anatomy. And they're also quite variable in between individuals, so it's hard to gather data from a lot of people and compare it to see what treatment approach works best for an individual AVM. So I hope to address this with an MRI imaging method called 4D Flow MRI. You may have had an MRI yourself, and it probably looked something like this. A picture of the inside of your head, and it might have also shown the shapes of the vessels, like this image here. But images like this one can't give us any quantitative information about the blood flow through the brain, and so even though AVM treatment is based on multiple sources of information, it doesn't include quantitative numbers that can give us a metric for how to treat the problem. So what I want to do is replace images like this, which is a cross-section shown by the yellow box on the left, by movies like this one. So this is an image collected with 4D Flow MRI, and the lines are showing the direction of blood movement. The colors show the velocity. So the red colored flow is fast, and you can see it the most in the arteries. And the blue colored flow is slow, so you can see it the most in the veins. But when we look at this kind of data from an AVM patient, what we see is that some of the red flow from the arteries that's really fast is going into this tangle of vessels, and then that's getting dumped right into the veins. So clearly, the AVM is changing the blood flow arrangement in the brain. And what I want to do is figure out how we can use this information to impact the treatment. And building this relationship between the imaging and the treatment actually requires a whole process. So we start with the clinical question of how does the AVM change the blood flow? And from there we need to decide how can we get that information efficiently. Once we get the information, we try to see how can we process the information and present it so that all the key facts about each vessel are clear from what we're showing. And then finally, we get feedback from clinical collaborators, and we try to revise this whole process so that we can really see, can this information impact the treatment decisions that physicians and patients are making? And for the rest of the talk, I want to take a deep dive into this section, which I think you may find some things to relate to. For example, suppose, I'm going to take a quick digression. For example, suppose it's 5 p.m. on a weekday and I want to get to the O'Hare airport. I would pull up a map on Google Maps, and if I'm looking at a satellite image like this, all the information I need to know is there, but it's so detailed that it's hard for me to make any conclusions from it. So what Google does is they simplify that information by removing some of the details, but highlighting the parts that are really important. And here we can see which things are connected to each other, like which roads connect us to O'Hare. And once we've done that, we can use that information to calculate a number that can summarize the situation for us. For example, it'll take us over an hour to drive to O'Hare, so we should probably take the blue line. Classic Chicago. In my research, the question is something like, how can we come up with a number that will describe an AVM? And what I want to do is start with the 4D Flow MRI information, which is so rich and complex that it's hard for me to generalize this between patients. But what I can do, and what I do in my research, is use a method of presenting the data called a flow distribution network graph, which looks something like this, to summarize what's really important. So in this method, what I do is I describe or represent each vessel as a straight line, and they're color-coded in this image by the amount of flow going through the vessel during a single heartbeat. And what this method does is really bring to the foreground which vessels are connected to each other and to the AVM. So by doing that, I can quickly identify the relationships between the vessels and use that relationship to calculate a number called the arterial to venous pulsatility index ratio. And you're gonna have to take my word for it that this number describes the relationship between arteries and veins. But what you can see in this graph over on the right, even if you blur your eyes and don't read the labels, is that one of these columns is very different from the others. And it turns out that that's the value of the ratio that describes arteries and veins connected to the AVM, as opposed to arteries and veins in healthy controls, as well as arteries and veins in the AVM patients that aren't actually connected to the AVM. So I think that this is a number that can help summarize what's going on in the AVM. And the future of this work is hopefully to see whether this number is related to the treatment outcome in some way and whether it can be useful to physicians trying to make these decisions. So we go from our way of representing the data to our way of quantifying the data to ultimately the answer to our question. So that work is still in progress, but what I hope to leave you with today is that the way you look at your data can really impact what you can learn from it. And I hope you find that as exciting as I do. If you're interested to learn more about the work our lab does, please visit our lab website at cvmri.northwestern.edu. And thank you for listening.